Hi, this is the lecture recording that we're calling the second segment of 3 segments on muscle. In the first segment, I covered most of the topics listed here. In the second segment, we'll finish up the topics listed here by talking about muscle metabolism as well as muscle fatigue. So when we talk about muscle metabolism, what should come to mind is all the ways in which muscle cells use ATP. And whenever a cell uses ATP, that cell also needs to be able to generate or replenish that ATP. So we're going to think about ways that muscle cells use ATP as well as ways in which muscle cells generate ATP. Muscle cells are some of the most metabolically active cells in our body. And by metabolically active, I mean there's a lot of biochemical processes that are happening in muscle cells all the time, and particularly during muscle contraction. And we've recently been studying the physiology of muscle contraction, and we talked about previously 2 separate processes that require muscle cells to spend ATP, and both of them have to do with muscle contraction. Can you think about 2 ways or identify 2 ways in which muscle cells in particular use ATP? Hopefully this image looks familiar to you. We went through it in quite a bit of detail in the previous lecture recording segment, in segment 1. And in this image, we can point out the 2 ways that muscle cells use ATP that are related directly to muscle contraction. The first way is shown here. Calcium pumps constantly use ATP to actively transport calcium out of the cytosol and into the sarcoplasmic reticulum. And these calcium pumps operate 24/7. They are always using ATP to ensure that the cytosolic concentration of calcium is low and the concentration of calcium inside the sarcoplasmic reticulum is high. The second way in which cells use ATP is during the contraction cycle. I'll remind you that during the contraction cycle, a molecule of ATP is needed for 2 reasons. That 1 molecule of ATP is needed to break cross bridges, and then that same 1 molecule of ATP is hydrolyzed, or it's split, in order to re-energize the myosin head so that myosin head will be able to go on and do another power stroke. So because muscle tissue has a high metabolic demand, it makes a lot of ATP and it uses a lot of ATP. It is supplied with the resources required for replenishing its ATP supply. So muscle tissue is well vascularized, and that means that there is a high density of blood vessels, particularly capillaries, in skeletal muscle tissue. And this is important because blood delivers the ingredients that are required to make ATP, including oxygen and glucose. Blood also carts away the waste products that are generated while making ATP, such as carbon dioxide. So here we're looking at a blood capillary that is sort of sandwiched in between 2 neighboring skeletal muscle cells. And in the skeletal muscle cells, we see that there's a banding pattern. Those are called striations, and those striations are caused by that regular overlapping arrangement of thick and thin filaments in the myofibrils. So it's cool that we can see that through a microscope. And the blood capillary that's sandwiched between those 2 cells is a very small blood vessel. An erythrocyte is a red blood cell. And you can see how small the blood vessel is because you can compare the diameter or the thickness, the width, I guess, of the lumen of the blood vessel to the width of a red blood cell. So capillaries are just a little bit larger than red blood cells, and the red blood cells need to pass through the capillaries in single file. And so, of course, those red blood cells are carrying oxygen. And as we will study when we get to the cardiovascular system and the respiratory system, there is gas exchange occurring as that blood passes through the capillary. As that blood passes through the capillary, oxygen is leaving the red blood cells and entering the muscle cells. And simultaneously, carbon dioxide is leaving the muscle cells and entering the blood. So we have this gas exchange that's occurring, and that oxygen is required for ATP synthesis, and that carbon dioxide is a waste product generated as a part of cellular respiration. When the cell is busy making ATP, carbon dioxide is created as a waste product. In skeletal muscle tissue, the capillaries have this really cool structure. They corkscrew, they spiral around the exterior of muscle cells. So muscle cells can contract, those muscle cells can get shorter, and they can relax, they can get longer without disrupting the structure of the capillary bed. And the capillaries are able to allow blood to flow through them. And as that blood flows through the capillaries, there's this constant exchange between blood inside the capillaries and the skeletal muscle cells. So a few slides back, we talked about two ways in which skeletal muscle cells utilize ATP, and this is unique to skeletal muscle cells, right? Skeletal muscle cells have sarcoplasmic reticulum, and there are calcium pumps that are constantly performing active transport to move calcium against a concentration gradient, to remove calcium from the cytosol and to put that calcium into the sarcoplasmic reticulum. And then again, during the contraction cycle, that a molecule of ATP is necessary to break cross bridges, and then that same molecule gets hydrolyzed or split, and that re-energizes the myosin head. And then number 3 is not unique to skeletal muscle cells, but then there are all the other ways in which cells in general utilize ATP. So muscle cells are constantly manufacturing proteins, they're constantly breaking down old proteins. Of course, they have sodium potassium pumps that are constantly functioning. Their cytoskeletons are constantly moving and being updated. So number 3 is just all of their metabolic activities, and these metabolic activities are in common with other cell types. So I hope you recognize this image. We looked at it earlier, very early this semester, when I introduced, I think, the concepts of metabolism, and we will cover metabolism in much greater detail later on this semester. But this is generally what's known as the ATP cycle. So I'll remind you that ATP is a very energy-rich molecule, and the bonds between neighboring phosphate groups in a molecule of adenosine triphosphate, those are very energy-rich bonds. And so when we, moving from the top red ATP, moving in a clockwise fashion, where it says phosphate removed, that means that ATP has been cleaved or hydrolyzed into ADP. So now we're at the bottom position in green. And that is an exergonic reaction that releases energy, and that energy can be used for cellular function. For example, that energy can be used for pumping calcium into the sarcoplasmic reticulum, or that energy can be used for re-energizing the myosin head. And so now we're down below at our green molecule of ADP, and that molecule of ADP needs to be recycled into a new molecule of ATP. In order to move in that clockwise fashion from ADP up to ATP, we need to add energy in order to reattach a third phosphate group. That is an endergonic reaction. It's an energy requiring reaction. And so where do we get that energy? That energy comes from, it's transferred from the chemical bonds in the food molecules that we eat to the chemical bond between neighboring phosphate groups in a new molecule of ATP. So when we talk about muscle metabolism, we're talking about the specific ways in which muscle cells make and use ATP. So here's that overall energy requiring reaction. We start with a molecule of ADP and we need to attach an inorganic phosphate. That's the P sub I, that just means inorganic phosphate. We need to attach those things to each other. That's an energy requiring reaction. We need to invest energy there in order to create a new molecule of ATP. So most body cells have at least two ways to perform this chemical reaction to attach that third phosphate. And we will talk about glycolysis, or splitting of glucose, and we'll talk about another process called oxidative phosphorylation. Most of our body cells are able to generate new molecules of ATP by these two separate processes. So glycolysis occurs in the cytosol of our cells, and it's called an anaerobic process because it does not depend on oxygen. In fact, the whole multi-step process does not require any oxygen. And then number two, oxidative phosphorylation. It's called oxidative because it does require oxygen in order to phosphorylate or attach a new phosphate group to a molecule of ADP to make ATP. And oxidative phosphorylation occurs inside of our mitochondria. And of these two processes, glycolysis and oxidative phosphorylation, definitely oxidative phosphorylation generates more ATP than glycolysis. So here's a visual representation of the process of glycolysis. On the left side of that diagram, we see a molecule of glucose where in gray, we have represented the 6 carbon atoms that make up a molecule of glucose, right? C6H12O6. So during this multi-step process, as that arrow proceeds from left to right, we see several things here. Number one, we see that that 6 carbon glucose molecule is ultimately going to be broken in half to form 2 3 carbon molecules of a substance called pyruvate, also known as pyruvic acid. So glucose is our starting product, is our starting molecule, and pyruvate is our product. And then in between there, during that process, the multi-step process called glycolysis, we will need to invest a couple of molecules of ATP. So on the right there, you see 2 molecules of ATP go into the process and 2 molecules of ADP come out of the process. So this is an energy investment. The cell is spending energy here. And then we also see that 4 new molecules of ATP are generated in the process of glycolysis. So now I'm looking sort of on the right side of the diagram, where we have 4 molecules of ADP are being input and 4 molecules of ATP are being generated as output. So this process of glycolysis requires an energy investment of 2 ATP, but it yields 4 ATP. So this is like you spending $2 to make $4. So what's your net profit? If you spent $2 to get $4, your net profit would be $2, right? So glycolysis generates just a little bit of ATP. And also I want to point out there in the middle, we have a molecule called NAD plus being converted into NADH, being reduced into NADH. That will play a bigger role for us when we talk about metabolism towards the end of the semester. So that's another way that energy can be harvested from this glucose molecule. So for now, please just know that glycolysis means splitting glucose, where 1 6 carbon glucose atom will be split to form 2 3 carbon molecules of pyruvate or pyruvic acid. And it does require a little bit of ATP to create this process, but we generate a little more ATP. And glycolysis does not require oxygen. It occurs in the cytosol of the cell, and it is a relatively fast process. In contrast, oxidative phosphorylation occurs inside the mitochondria. It does require oxygen. It is a multi-step process, and therefore it is slower than glycolysis, but it generates far more ATP than glycolysis. So in glycolysis, if we had a net gain of 2 ATP per molecule of glucose, oxidative phosphorylation yields us a net gain of 32 ATP per molecule of glucose. So we get a much bigger yield, but it takes more time, it takes more resources. So here's where we get into the parts that are unique to muscle cells. Most cells perform glycolysis and oxidative phosphorylation. Muscle cells have this third trick for producing new molecules of ATP, and that is because they have a particular substance called creatine phosphate. So creatine phosphate is a small amino acid-like molecule, and it's produced in our livers, and it travels in our blood, and our skeletal muscle cells can collect creatine phosphate. So now it's inside of our skeletal muscle cells. And creatine phosphate is a muscle cell's way of storing or stockpiling phosphate groups, these high energy phosphate groups. While the muscle cell is at rest, because the muscle cells have creatine phosphate, they're able to sort of accumulate extra phosphate. And then when that muscle cell starts to work vigorously because the contraction cycle is happening, that stockpile of phosphate can very quickly be used to generate new molecules of ATP. Here we see a bidirectional chemical reaction where creatine is converted into creatine phosphate. Creatine's on the left and creatine phosphate's on the right. Another word for creatine phosphate is phosphocreatine. It doesn't really matter. So on the left side, we just start with creatine over here, and then on the right side, notice over here we've added a phosphate group. And the artist took care to represent that chemical bond as a high energy chemical bond. Notice the squiggly line. So where did that phosphate group come from, and why is there a high energy bond there? Notice that in order to convert creatine to creatine phosphate, we need to spend a molecule of ATP. And so that high energy phosphate is transferred away from a molecule of ATP and onto the creatine to generate creatine phosphate. And what are we left with? We're left with the spent molecule of ADP. So when we want this reaction to go from left to right, we are transferring a high energy phosphate group away from a molecule of ATP and onto creatine to generate new molecules of creatine phosphate. And this reaction from left to right happens when our muscle cells are resting. When our muscle cells are resting, there's not a particularly huge demand for ATP, and so our muscle cells have the opportunity stockpile these phosphate groups in the form of creatine phosphate. The reaction will proceed in the opposite direction during muscle contraction. During muscle contraction, this phosphate group here can be very quickly tacked onto this ADP in order to make a new molecule of ATP. So when the reaction proceeds from right to left, we very quickly utilize that high energy phosphate group that is part of creatine phosphate to generate new molecules of ATP. And this process is very quick, and it happens while the contraction cycle is on in our skeletal muscles. This is a different artistic representation of exactly the same concepts. So in our muscle cells all the time, we have a molecule called creatine. When our muscle cells are at rest, we take existing ATP and transfer that high energy phosphate to produce new molecules of creatine phosphate. And so in this way, while our muscle cells are relaxed, we are using ATP to stockpile phosphates for the future. And then when our muscle cells are contracting and our muscle cells are very quickly depleting the cell's supply of ATP, this creatine phosphate provides a quick way of attaching a phosphate group onto an ADP to make new molecules of ATP, and this new molecule of ATP can go on to support the contraction cycle, or maybe it'll be used to continue to pump calcium ions into the sarcoplasmic reticulum. So here we can put together visually the three ways that muscle cells can produce ATP. So the first two, glycolysis and oxidative phosphorylation, these two are not unique to muscle cells. Most of our cells are able to split glucose and derive a little bit of energy from that. And where we see that energy from glycolysis is ATP up here. And then most of our cells are also able to use the products of glycolysis. Notice this arrow here. This is the pyruvic acid, or the pyruvate. Is used in oxidative phosphorylation, and that also produces ATP. And then this is the thing that's unique to skeletal muscle cells, creatine phosphate. Our muscle cells are able to stockpile those phosphate groups, and then during muscle contraction, when there's a high demand for ATP, this creatine phosphate can be depleted back down into creatine, but that phosphate group is attached to this molecule of ADP to create a new molecule of ATP. So notice that there are three arrows in this image pointing to new molecules of ATP in the center there. And these are the three processes by which skeletal muscle cells can generate new molecules of ATP. Muscle cells have this highly variable demand for ATP. When your muscle cells are at rest, the demand is relatively low. And as soon as your muscles become active, the demand for ATP skyrockets. And so creatine phosphate is there to ensure that there is an adequate supply of high energy phosphate groups to allow muscle contraction to continue for at least the first 15 seconds of muscle activity. So let's think about your gastrocnemius for a little bit. That is a muscle in your calf. It's the largest muscle in your lower leg. And so maybe you're sitting down right now. If you were to get up and walk and go and get yourself a glass of water, for that initial new skeletal muscle activity, the creatine phosphate would be completely depleted down to creatine in about 15 seconds of muscle activity. And so it's important to consider evolution of human physiology. And for most of human evolutionary history, there were dangers about. And so that 15 seconds of security in terms of ATP production in our muscle cells, that 15 seconds will be enough for you to scramble up a cliff before falling. It might be enough for you to climb a tree and avoid a predator. But after that 15 seconds, our muscle cells have depleted our supply of creatine phosphate. And all of those creatine phosphate molecules have now been converted back into creatine. And creatine doesn't really have anything else to offer during that walk over to the sink to get a glass of water. So perhaps you've heard of creatine supplements. You can order them online or go into any vitamin type store and buy creatine supplements. Are they effective? That's a great question. What does the research show? So we know that creatine, this small amino acid-like molecule, we know that that is manufactured in your liver cells, and we can also obtain creatine as a nutrient from eating food. Right? Creatine is part of muscle cells, and muscle cells are part of meat. So you can augment your dietary intake of creatine by eating meat. We can also augment our dietary intake of creatine by taking a pill, by taking a creatine supplement. So do they work? And in the scientific literature, there's an existing controversy here. Some studies support the hypothesis that creatine supplementation does improve athletic performance, but particularly during explosive movements, such as sprinting and weightlifting. And then simultaneously, there are other studies that do not support that hypothesis, that creatine supplements have no effect on athletic performance, including explosive movements. And so there is really no scientific consensus on the efficacy of creatine supplements. Do they enhance athletic performance or not? There's a conflict in the scientific literature. But there is concern because the layperson might believe that creatine supplements do, in fact, enhance athletic performance. There is some concern that people will overdo it. And we're not certain of the feedback mechanism. So when somebody supplements their body with creatine, what happens to creatine synthesis that normally occurs in the liver? Is that downregulated? Is that not downregulated? We do know that creatine supplements at higher than a recommended dose put both the kidney and the liver at risk of being damaged. So if you or someone you know, someone you love is taking creatine supplements, be sure that they are adhering to the recommended dosage. So I just told you about one trick that muscle cells use for being able to constantly replenish their ATP supply. Muscle cells have a whole bag of tricks. In addition to creatine phosphate, muscle cells are also able to sort of keep a pantry or a supply closet full of the necessary ingredients for generating ATP. So one of those necessary ingredients is phosphate, and muscle cells can store or stockpile phosphate in the form of creatine phosphate. Another one of those ingredients required to make ATP is glucose, and muscle cells can stockpile or store glucose in the form of glycogen. And then a third ingredient that's required to make ATP is oxygen, right? That's going to feed into oxidative phosphorylation. And so a red pigment called myoglobin that is a muscle cell's way of temporarily storing or stockpiling oxygen. We've seen this picture before when we talked about the physiology of muscle contraction, and at the time I ignored some components of this image, and I want to circle back to this image so that we can focus on these glycogen. Let me choose a different color. We can focus on these glycogen granules in the cytosol. We can talk about myoglobin in the cytosol. We can talk about how muscle cells have many mitochondria. So creatine phosphate is not shown here, but that also is found in the cytosol of a muscle cell. Glycogen granules, I'll remind you that glycogen is a polysaccharide where the individual units are molecules of glucose. And those molecules of glucose have been tacked together to form this big polysaccharide called glycogen. And the myoglobin, myo means muscle, myoglobin. Myoglobin is a reddish pigment. It's very similar to hemoglobin, and it can temporarily store oxygen molecules inside the cytosol of a muscle cell. Uniquely in muscle cells, the ingredients for ATP synthesis are stockpiled or stored in the cytosol. Creatine phosphate is the source of phosphate, glycogen is the source of glucose, and myoglobin is the source of oxygen. And these are unique to muscle cells. Here we're looking at an image of glycogen. Glycogen is a polysaccharide, so that's a carbohydrate made out of many subunits, where the subunits, the monomer building blocks, are glucose molecules. And glycogen is quickly and easily cut up or catabolized into individual glucose molecules. This does happen inside your skeletal muscle cells, and that those individual glucose molecules are used as fuel for ATP production. Myoglobin is similar in structure to hemoglobin, which we'll talk more about when we get to the cardiovascular system. Both myoglobin and hemoglobin are metalloproteins that can reversibly bind oxygen. A metalloprotein is a protein that includes metal, where the metal here is iron. So when the muscle cell is relaxed and not busy contracting, myoglobin can accumulate oxygen so that during muscle contraction, that oxygen is on hand inside the cytosol attached to myoglobin, and it can be released from myoglobin in order to support oxidative phosphorylation. So we can examine different types of meat, from white meat to dark meat. And what makes them vary in color, for the most part, is myoglobin content. So because myoglobin stores oxygen, it supports oxidative phosphorylation. It doesn't support glycolysis. Remember, glycolysis does not require oxygen. And oxidative phosphorylation that occurs inside the mitochondria is responsible for the vast majority of ATP production, particularly for ongoing long endurance muscle activity. So muscles that are typically involved in burst activity, so we just need a quick burst of power and then the muscle can stop working, those muscles tend to be very light in color because they have little myoglobin and they have a lower vasculature, reduced vasculature, not as many blood vessels. And muscles that are dark in color because they have lots of myoglobin to store lots of oxygen, and they also are highly vascularized. They have lots of blood vessels that can also deliver lots of oxygen. And so the oxygen demands on a particular muscle vary from muscle to muscle, and some of our muscles are typically required or play a role in short burst activity, like our arm muscles, for example. For most of human evolutionary history, our arm muscles were used for things like grabbing and pulling and punching and throwing, but our leg muscles carry us around all day long. Humans, from an evolutionary perspective, we are built for walking and running. So there is more myoglobin in our leg muscles than in our arm muscles. And we can think about that through the lens of the foods that we eat. So to me, it looks like these three pieces of meat from left to right come from a fish, a white fish, which is sort of a generic word for a type of fish that we eat, that is consumed, that is very light in color. And then in the middle there, that sort of looks like chicken breast, although maybe arguably it's a type of salmon, but I think it looks like chicken breast. And then on the right, that looks like beef, or maybe venison, maybe it's deer or bison. So we're going from light to dark in these meats, and light corresponds with no myoglobin. So those muscles there are not very long endurance muscles. Those are muscles that are responsible for very short bursts of activity, perhaps to escape danger or to for a brief stint required to prey. And then all the way on the other end of the spectrum, lots of myoglobin. On the right side, that dark meat is those muscles from whatever part of the body they come from in that organism. Those muscles work a lot all day. They need to keep going. They don't fatigue easily. They keep producing ATP, and one of the ways that allows them, one thing that allows them to keep producing ATP is that those muscles can store oxygen as attached to myoglobin. And then the image in the middle has sort of a moderate amount of myoglobin. So those muscles there in the middle, maybe they're chicken breast muscles, not sure. Those muscles can have a moderate endurance. So in this image, we see summarized the three forms or the three processes that allow for ATP synthesis in muscle tissue. So in panel A, we see what's known as direct phosphorylation. So that phosphate group is transferred from a molecule of creatine phosphate directly to a molecule of ADP to make ATP. And then in the middle, panel B, is glycolysis. Glycolysis is also known as anaerobic cellular respiration. And in that anaerobic pathway, we split glucose. We invest 2 molecules of ATP. We produce 4 molecules of ATP, so our net gain, our profit, is 2 molecules of ATP. And this is a relatively fast process. It does not require oxygen, and it occurs in the cytosol. And then finally, panel C on the left, that's known as the aerobic pathway. I've also referred to it as oxidative phosphorylation. So it does require oxygen. And the products of glycolysis, pyruvate or pyruvic acid, same thing, pyruvic acid becomes the starting ingredient for aerobic cellular respiration that occurs inside the mitochondria. And maybe you've studied this before, and we will study it later on, but inside the mitochondria, we will find 2 important processes. One of them is the Krebs cycle, and the other is the electron transport chain. So that's what comes ahead for us. But for now, please understand that the aerobic pathway occurs inside mitochondria. It is a slower pathway because there are many more steps involved. It does require oxygen. It does produce many more ATP than the other 2 processes shown here in panel A and panel B. And the aerobic pathway is most important for sustained muscle activity. So more than a minute of muscle activity, and the vast majority of that ATP is coming via the aerobic pathway. Here's an important concept, and I'll just read it from the slide because it's that important. Endurance activity is limited by the capacity of the cardiovascular system to supply enough oxygen to support aerobic cellular respiration. So what does that mean? Endurance activity, so this ongoing muscle action. So it's not a burst activity, it's not sprinting, it's not kicking, it's not punching, it's walking around all day, for example. So endurance activity is limited by the capacity of the cardiovascular system to supply enough oxygen. What does that mean? The ability of the cardiovascular system to supply enough oxygen. So we have not yet studied the cardiovascular system, but one of the functions of the cardiovascular system is to pick up oxygen in our lungs and to transport that oxygen to our body tissues. And here, the tissue that we're focused on is skeletal muscle. So, endurance activity, this ongoing muscle action, is limited by the ability of your cardiovascular system, of your heart, blood, and lungs to supply enough oxygen to support oxidative phosphorylation in the mitochondria of your muscle cells. We're going to stick with this concept for a couple of slides. What that means is when there is insufficient oxygen delivery to your muscle cells, and by insufficient I mean there's not enough oxygen delivered to support the full oxidation of glucose. So that pyruvate, that's the product of glycolysis, it enters the mitochondria, and if there's not enough oxygen being supplied to the cell, then that pyruvate cannot be further oxidized to yield more molecules of ATP. So there's still a lot of energy in that pyruvate molecule, and rather than just waste it or let it hang out in the cell, that pyruvate, that energy-rich molecule, gets converted into lactic acid. And lactic acid is a small, fairly lipid-soluble molecule. And so it pretty quickly diffuses out of the muscle cell into the blood, and it travels in blood, and it specifically gets taken up by your hepatocytes or your liver cells. And it's a valuable resource. There's still a lot of energy in that molecule. So your liver cells take it up, and one way or another, it gets reused to make ATP, either by your liver cells or it might get converted back into glucose, actually. We can use this image to put many recent concepts together. And I guess I'll just start at the beginning And there's not a lot of green on this image, so I'll choose green to start with. So let's just start at the top, except that's yellow. Hold on. Let's just start at the top with muscle glycogen. So I hope you remember that glycogen is a polysaccharide, and it can be catabolized right there in the cytosol of the muscle cell. It can be cut up into pieces, and those individual pieces are units or molecules of glucose. And then glucose is, of course, the starting ingredient for glycolysis. So inside a skeletal muscle cell, that glucose can come from one of two places. It can come from the pantry, right? The storage closet inside a muscle cell. Muscle cells have glycogen in them, and that glycogen can be liberated to form glucose, and then that glucose can be used for glycolysis. The second source is from our cardiovascular system. So there is always glucose in your blood. You need to have that glucose in your blood or else you die. And that glucose in your blood can enter body cells, including muscle cells, and from either source, whether that glucose came from muscle glycogen or whether that glucose came from the blood, that glucose is your muscle cells' preferred starting ingredient, really, for making new molecules of ATP. And that process at this point is called glycolysis. And you remember that there is a net gain of 2 molecules of ATP per molecule of glucose split during glycolysis. And glycolysis results in addition to producing 2 ATP, glycolysis results in the formation of 2 molecules of pyruvate, also known as pyruvic acid. And this is still an energy-rich molecule, and if there is sufficient oxygen being delivered to the muscle cell, then these molecules of pyruvic acid, you know where they go? They go into the mitochondrion. Yeah, and so a shorthand for mitochondria is lowercase m, lowercase t, means mitochondria. However, if there is insufficient oxygen to further support the oxidation of pyruvic acid, the pyruvic acid molecules do not enter the mitochondria, but rather, right there in the cytosol, the pyruvic acid undergoes a short biochemical process and is converted into lactic acid, which is also known as lactate. And this is it's a small molecule, it's a very simple molecule, and it contains actually most of the energy that was in that original molecule of glucose. We got a little bit of that energy out when we have a net gain of 2 ATP, but most of the energy in that original molecule of glucose is present in these 2 molecules of pyruvic acid, and if that pyruvic acid does not undergo oxidative phosphorylation in the mitochondria, if there's not enough oxygen being supplied to the cells to support that, then those 2 molecules of pyruvic acid are converted into 2 molecules of lactate or lactic acid, and that is not used in the muscle cells immediately. Rather, lactic acid is a small lipid-soluble molecule. It diffuses out of the muscle cell into the blood, and once it's in the blood, it gets taken up by liver cells, and those liver cells can either use lactic acid to make ATP, or they can convert that lactic acid back into glucose. They can store that glucose as glycogen so that energy is not lost. We're back at that concept that I read to you off of the slide earlier, and it's so important that I will read it again. Endurance activity, so prolonged muscle activity, is limited by the ability of the cardiovascular system to supply enough oxygen to support oxidative phosphorylation in the mitochondria. So I hope we have all had this experience that you engage in some type of endurance activity. My favorite endurance activity is dancing, but maybe you've done rowing or swimming or running, and after so much activity, you're huffing and puffing and you need to take a break. Well, why do you need to take a break? And why does maybe your friend not need to take a break? What's the difference in physiology between you need to take a break, but your friend can just keep going and going and going? The difference is your body's ability to deliver oxygen from your lungs to your muscle cells, and that oxygen is required to support aerobic cellular respiration. So there are athletes, world-class athletes, that spend their whole lives training to be the best in their field. They compete very intensely. Why does one person win and another person lose? It is determined, for the most part, by the ability of their cardiovascular system to supply oxygen to their muscle cells. That's a crazy idea, but it's supported by lots and lots of evidence. So, one reason, or the reason I should say, why endurance athletes, marathon runners, Olympic marathon runners, the reason why they train at high altitude is because life at high altitude increases red blood cell production. And our red blood cells are the oxygen carriers in our cardiovascular system. The more red blood cells you have, the greater the capacity for oxygen transport in your cardiovascular system. So let me take a step back. Why does a high altitude environment increase red blood cell production? We'll get to that more so when we get to the respiratory system. But I'll tell you right now that at higher altitude, we're further away from the core of the planet Earth, and the air molecules get further away from each other. So there is a lower, it's called partial pressure. There's a lower partial pressure of oxygen at high altitude than there is at sea level. And for right now, you can actually think about it in terms of density. For every particular volume of air inhaled, how many oxygen molecules are actually inhaled? We inhale more oxygen when we're breathing at sea level than we are breathing at, say, 10,000 feet. So, in response to a lower partial pressure of oxygen in the atmosphere at high altitude, our cardiovascular system, our bone marrow specifically, produces a higher number of red blood cells. A higher number of red blood cells will enable our cardiovascular system to better obtain oxygen from our relatively low oxygen atmosphere at high altitude. And this is why athletes, endurance athletes, often train at high altitude, because it increases their red blood cell production. It increases their hematocrit. So somebody who has trained at a high altitude has a higher proportion of red blood cells than somebody who has trained at low altitude. And then another strategy for augmenting or enhancing the capacity of your cardiovascular system to deliver oxygen is called blood doping. And if you pay attention to any type of endurance professional sport, you're familiar with the term blood doping. It comes in a couple of varieties, but what they have in common is blood doping is a technique for artificially increasing the oxygen carrying capacity of blood. So we're going to artificially increase your hematocrit, or the proportion of blood that is made up of red blood cells. So two ways. The old school form of blood doping is number one here. So several months before the endurance event, several months before the Olympic trial, for example, the athlete removes some blood from their body, puts it in the freezer for later use. They continue to train, and then right before the event, they deliver that stored blood back into their body. So they have artificially increased or augmented the number of red blood cells in their body. And then number two, that's a newer invention, and then there are all sorts of forms of this, where hormones that synthesize, so hormones that signal to cells that make up red bone marrow, these hormones cause the cells in red bone marrow to produce more red blood cells. And that hormone, the primary hormone that's responsible for that, is called EPO. And we'll talk a little bit more about EPO when we get to the cardiovascular system and the urinary system, because it's actually produced by your kidneys. So we can inject hormones to increase the synthesis of red blood cells, and both of these forms of blood doping artificially augment or enhance the oxygen carrying capacity of blood. So muscle fatigue. I hope we have all had this experience as well. You've been working your muscles for a certain amount of time, and at some point they stop, your skeletal muscles stop behaving the way you'd like them to. So there is a weightlifting technique called work to failure. And of course, that's the layperson's technique. But if you spend time in a gym lifting weights and you're at all interested or pay attention, you're familiar with that term, work to failure. And so what does that mean? That means maybe you're doing bicep curls with a particular weight level. Let's say you're using a 20-pound weight to do bicep curls. And you do as many bicep curls as you possibly can. And at some point, your bicep is going to, and I put air quotes around this, your bicep is going to refuse or not be able to lift that weight anymore. And that's due to muscle fatigue. So your central nervous system is sending the commands, lift the weight, lift the weight, lift the weight. And the muscle is just no longer able to respond. So muscle fatigue is defined as the decline in the ability of a muscle to maintain contraction after prolonged activity. It's a really neat thing to observe in your own body. And we have somewhat of an understanding of muscle fatigue, but our understanding is definitely far from complete. So let's think about some of the factors that contribute to muscle fatigue. I hope you have a pretty thorough understanding of what goes into, what biochemical factors are required to produce muscle contraction. And we can take a look at that process and see, oh, well, what might go wrong to produce muscle fatigue? Not wrong, but what might be disrupted that leads to muscle fatigue. So we can start at the neuromuscular junction. So let's stick with this example of doing bicep curls, right? You've been doing bicep curls, bicep curls, bicep curls, and every time you do a bicep curl, several motor neurons release acetylcholine at the neuromuscular junction, and after so many releases, those motor neurons might start to be depleted in their supply of acetylcholine. So there might be what plays a role in muscle fatigue is reduced stimulation or reduced signaling between the motor neuron and the skeletal muscle cell due to insufficient release of acetylcholine. Our muscle cells can also deplete their supply and their capacity for regenerating ATP. So as we talked about recently, in the first few seconds of muscle activity, our cells' supply of high-energy phosphate is rapidly depleted as creatine phosphate is converted into creatine. And then our cells' on-hand supply of glucose in the form of glycogen gets depleted as that glycogen is catabolized or chopped up, and that resulting glucose is used to produce ATP. And then we have oxymyoglobin. Oxymyoglobin means it's myoglobin that has an oxygen attached to it. And as our muscle cells continue to use oxygen to produce new molecules of ATP through oxidative phosphorylation, that oxymyoglobin gets depleted just into myoglobin that no longer has any oxygen attached. Another factor that contributes to muscle fatigue is a reduced release of calcium from the sarcoplasmic reticulum. So as you, I hope, remember, with every action potential that travels down a T-tubule, that causes the release of calcium from the sarcoplasmic reticulum. Well, none of our membranes are perfect. And every time calcium is released from the sarcoplasmic reticulum, a little calcium leaks out of the cell across the plasma membrane. So release after release after release, there will ultimately be reduced calcium inside our sarcoplasmic reticulum and reduced calcium release into the cytosol with every single muscle contraction. And you, I hope, remember the role that calcium plays in the physiology of muscle contraction. Calcium binds to troponin, which is required for revealing the myosin binding sites on actin. And then another contributor to muscle fatigue is this accumulation of lactic acid. When the cardiovascular system is not able to supply enough oxygen to fully support the complete oxidation of pyruvate, then some of that pyruvic acid or pyruvate does not enter the mitochondria, but it's still this energy-rich molecule, and it gets converted into lactic acid. And there can be just like this super short-lived temporary buildup of lactic acid, also known as lactate. It's a small lipid-soluble molecule, so it readily leaves the cell, and it enters blood and gets picked up by your liver cells. But for this temporary buildup, this lactic acid, it's an acid, so it contributes hydrogen ions to a solution. That's the definition of an acid. And so this super temporary buildup of lactic acid can actually lower the pH of the cytosol. And I hope you remember that pH influences the three-dimensional shape of proteins, and that three-dimensional shape of proteins influences their function. So by temporarily lowering the pH of cytosol, we can reduce the activity of proteins, enzymes, as well as contractile proteins. So this is a really great video to watch. It's actually hilarious to watch it in a classroom context. So this video shows a minute and a half of the end of an Ironman competition. So the Ironman is an athletic event, and it's a triathlon. So there are 3 different athletic events all rolled into one. So the competitors swim for 2.4 miles, then they ride a bicycle for 112 miles, and then they run a marathon. It sounds crazy to me, but believe it or not, there's often a student in the class who has performed an Ironman triathlon in the past. And so the participants have 17 hours maximum to complete all of these events. So I'll encourage you to go to YouTube. This link is in the supplemental resources in Canvas. Go to YouTube, it's a minute and a half, and I'll encourage you to look for evidence of muscle fatigue. It's obvious, but think about the muscles that are involved in running. So that's the last event. These people they've already swum for 2.4 miles, they've already biked for 112 miles, and now they are finishing up a 26-mile run. So think about the muscles that are involved in just running, period. So you're going to think about the quadriceps and the hamstrings and the gastrocnemius and the gluteus maximus and medius and minimus. And look at the muscles that these people are using just to cross the finish line. Because their larger muscle groups, like the quads and the hamstrings and the glutes, are so fatigued, the smaller muscle groups tend to be the ones that are needed to propel them toward the finish line. So the adductors and the abductors are used to help propel these people across the finish line. And so it's a terrific example of muscle fatigue. And that brings us to the end of segment 2 of 3 segments on muscle tissue. Thanks for watching.